This diagram of the mature adult urinary system shows a complex physiological structure required for filtering blood and maintaining human survival. However, the embryo does not build this final system directly from the start. The definitive human kidney is the final iteration in a sequence of 3 overlapping systems. As this timeline illustrates, development progresses through 3 distinct stages: the pronephros, the mesonephros, and the metanephros, appearing sequentially from week 4 into week 5. These systems develop in a strict cranial-caudal wave, moving from the head toward the tail down the embryo's body. Understanding the final anatomy of the kidney requires tracking the temporary embryonic scaffolds that preceded it. This developmental timeline begins at embryonic week 4, immediately following gastrulation. This cross-section of the embryonic disc identifies the 3 primary germ layers, with our focus resting on the mesoderm. We are narrowing our focus to the intermediate mesoderm, situated between the paraxial and lateral plates. This 23-day longitudinal view shows the intermediate mesoderm differentiating into the urogenital ridge, composed of segmentally organized buds along the posterior abdominal wall. This single ridge of tissue holds the blueprint for both the ultimate waste management system and the entirety of the reproductive tracts. The pronephros is the first prototype, appearing early in week 4 as the most cranial part of the system. It is located high in the cervical region of the embryo. In humans, this system is entirely vestigial and non-functional, degenerating almost as soon as it appears. As the pronephros fades, the second stage, the mesonephros, activates immediately. The mesonephros is located further down the body, in the thoracolumbar region. This middle panel shows that the mesonephros forms a functioning temporary kidney, utilizing defined excretory tubules that connect to the descending mesonephric duct. The embryo constructs this large, functioning thoracolumbar kidney with its own system of tubules and ducts, only to destroy it within weeks. By the end of the 2nd month, the main body of the mesonephros disappears. As the highlight indicates, the blue mesonephric duct is preserved due to its critical proximity to the developing gonads. The ultimate fate of this duct depends entirely on the embryo's chromosomal sex. In the female developmental pathway shown on the left, the mesonephric duct largely degenerates, leaving behind only minor remnants. In the male pathway on the right, the mesonephric duct is hijacked and repurposed to become the vas deferens and epididymis. The temporary excretory tubules are adapted to become the outflow tracts, connecting the testes to the duct. This makes the male reproductive tract a modified remnant of a temporary embryonic urinary system. The metanephros, or permanent kidney, begins development in the pelvic region during week 5. Its formation requires a critical biological trigger, the appearance of the ureteric bud. This diagram shows the ureteric bud sprouting from the caudal end of the mesonephric duct. The bud branches repeatedly to form the entire collecting system, including the renal pelvis, calyces, and collecting ducts. The ureteric bud then penetrates a second tissue mass, the metanephric blastema. This penetration induces the blastema to form the functional nephrons, including the glomerulus and Henle's loop seen here interfacing with the collecting duct. The metanephros glides upward from the pelvic cavity to its permanent position in the upper lumbar region over several weeks. During this ascent, the kidney undergoes a 90-degree medial rotation to position the hilum correctly. Fetal kidneys produce urine primarily to contribute to the amniotic fluid, while the placenta filters waste from the blood. The permanent kidney functions perfectly because these 2 distinct tissues, the bud and the blastema, successfully fused. What is the clinical result if the precise timing of this ureteric bud interaction fails? If the ureteric bud fails to form or degenerates early, it leaves a blank space where the kidney should be. Without the bud, the metanephric blastema is never induced to form tissue, resulting in renal egenesis. Alternatively, the ureteric bud may split prematurely before reaching the metanephric blastema. These duplicated buds induce 2 separate areas of tissue, creating a double kidney or duplicated ureter. These gross anatomical variations serve as direct evidence of the underlying embryonic sequence playing out incorrectly. The urinary system is built, destroyed, and rebuilt through the pronephros, mesonephros, and metanephros stages. The embryo repurposes the temporary mesonephric scaffolding to construct the reproductive outflow tracts. Ultimately, the mature adult human form represents the living, breathing history of these incredibly precise, beautifully timed embryological phases.